Dino Stock, I'm a medical doctor by training with a background in systems neuroscience and machine learning algorithms. And what we try to do in my group is what I will illustrate in 3 projects, and that is to reframe classical questions that we had in systems neuroscience, reformulate them from a pattern learning perspective, and try to translate solutions back to the application domain. But before, I will try to raise some awareness on prediction not being the same as statistical inference. So I think that there is increasing tension between more classical notions of importance in data analysis, such as we do it in classical linear models, and more modern or more frequently used types of analyses, such as those from machine learning. So what you see here is a bunch of gene expression fake data analyses, and we generated a bunch of data sets and then analyzed the same data that look like gene expression data in the most common way to distill statistically significantly important variables in panel A on the left, and then submit the same data sets to, again, the question of which of these genes are important, but based on a machine learning algorithm, in this case, random forests. So the important conclusion from this introductory slide is on panel C. There is not complete congruence between which types of genes in these synthetic data are identified to be relevant. So the conclusion is there may be a divergence between notions of importance in different statistical communities that may have been less in touch before. So we try to go into detail with these divergences in collaboration with people from Friday Lab Paris. What you see here in panel A on the left is the results from more than 100,000 empirical data simulations that vary in things that we typically care about, such as how many input variables do I actually have, how many samples, how many participants do I have, are there nonlinear effects or not, is there redundant information in my variables about the outcome that I try to predict, and so on. In this hexagonal binning plot, you see in each location how many of these more than 100,000 data sets ended up in a particular relationship between classical statistical significance on the x-axis and out-of-sample prediction accuracy, a gold standard to derive robust conclusions from data in the machine learning community. So, and this is zoomed in panel B on your right, where you see the more common p-values that we use in our community. And we were surprised to find that in many, perhaps most of these more than 100,000 fake data sets, we find some variables that were statistically significant. However, these models varied in the explained variance in new data that had not been seen by the model, because that varied between 0 and 100%. On the other hand side, those linear models, at least as we analyzed it here, that showed a good out-of-sample prediction performance and explained variance in data from subject we hadn't seen yet, those were more likely to also show some variables with a p-value that was evaluated to be statistically significant. So another distinction related to what I just said is the difference between interpretability and model complexity that we already have alluded to before. And what we try to plot here is how different classes of data analysis models or regimes map onto how well can I actually make circumscribed statements about particular input variables as to how relevant they are as to what I'm trying to estimate, and how complex can the relationship in data actually be that's on the x-axis that I try to predict and reproduce in new data. So what you see on the top left is linear regression or generalized linear types of models, which have been dominant in our community. So they are perfect in interpretability, but they are constrained because the representational capacity is limited. At the other end of this extreme is deep neural network models on the bottom right. So they can learn particularly complex relationships from data, but it's not clear at this point how well we will be able to fully understand the representations that they learn from data. So I'd like to emphasize that there are some exceptions to perhaps this interpretability, predictability trade-off that include generalized additive models and random forest algorithms that, in my subjective opinion, are underused at this point in our community. So the first project is on the default mode network, which is one of the most important networks in the human brain because it consumes so much energy, probably more than many, perhaps most other parts of the brain, and it's at the intersection of all these different major psychiatric disorders. So what we did here in 10,000 people from the UK Biobank is we tried to bridge, based on an atlas that we established before, variability in subregions of this major brain network, which you see on the right, and we tried to use this variability in gray matter to predict the variability of microstructural properties in white matter fiber tracts. So among 48 candidate white matter fiber tracts, the three most predictable tracks with up to 25% explained variance in fresh data, the top three were all parts of the fornix. So the fornix is a major output tract of the hippocampus. It is the backbone of the limbic system, and we speculated that perhaps some of the exciting research that's going on in invasive animal research on space and time cognition may perhaps indicate that this information may partly travel through the fornix to the higher association or cortex, including the default mode network. So we also wanted to know, okay, how about brain function, and how does functional coupling between subregions in the default mode network on the top, how is that systematically linked to how major brain networks, as we discover them using ICA on the bottom and C, how are those actually related to each other? So in here you see that it's not the entire nodes of the default mode, it's specific subregions in those that are most relevant in explaining variance in the communication of major brain networks, such as dorsal attention, saliency, and so on. So the most explanatory mode of population variation is this one here. So just to reiterate, we have functional coupling between subregions inside of the default mode network on the left. So it is data on how subnodes inside of the default mode network are communicating with each other. So a notion of default mode segregation. And on the right, we have how are functional ICA components connected to each other in 10,000 people. So what we find is that specifically the interior right and left TPJs, that those, if they connect strongly to pretty much anything else in the default mode network, that this pattern is coupled with explaining most variance in how major brain networks that cover the cortical mental, how this actually comes about. So we try to go more into detail and find out, is that really the TPJ where this influence may be coming from? So we performed a sensitivity analysis, lesioned and removed parts of the data to come up with these measures of population variability using bootstrap analysis to confirm that, as you see on the bottom left, that it's really the interior right TPJ that may be most relevant in what we speculate this part may do for mediating between network configurations to solve tasks in the environment. So we then wanted to go on and look at this from the genetic level. So this is in collaboration with the Broad Institute at Harvard MIT. And here you see genome-wide statistically significant locations, and we found dozens of those. However, the default mode features of the brain and aspects of human cognition are probably polygenic. So they're probably observed by large collections of genes rather than small groups of easily interpretable gene locations. So this is why we ran, again in 10,000 people, a genetic correlation analysis. And what you see here is how similar is the genetic control that controls the variability in the volume in parts of the default mode network. How are those sets of genes similar to how the volume in a different part of the default mode network is actually, how this comes about. So what you see here is that on the bottom, the dorsomedial prefrontal cortex is most different in what you would expect to be a coherent brain network. And we think this is exciting and promising because some people believe that Brodmann area 9 may perhaps be the most complicated part of the human brain and may be particularly relevant in the human-defining types of thinking that the default mode network is involved in. So another exciting analysis that you can run once you increase the sample size to these numbers is you can ask whether the profile of relevance across the entire genome, 14 million locations in the brain, whether those are similar overall, this is why it's a polygenic approach, to, for example, personality traits that have already been analyzed by consortia before. So what I just wanted to briefly mention here is that on the left you see how similar is the genetic control for volumes in different parts of the default mode to the personality trait openness. And on the right you have consciousness. So if you're a default mode researcher, you're enticed to perhaps think, well, that may be a difference between exploration on the left and how genetics relate to inter-individual variability in brain volume, and on the right, that may be more a marker of exploration, and that fits nicely these ideas that the default mode may be related to creative thinking. So the last project I just very briefly wanted to mention is upcoming work with that is actually based on a previous paper from Daniel Lopez from my lab, and we tried to establish a social brain atlas, integrating data from 4,000 experiments from 28,000 participants, and we came up with 36 target regions that indicate locations of highest neural activity changes across the brain in any types of social affective processing as we can measure it with fMRI. So, and with current PhD student Hannah Kieso and Robin Dummer from Oxford, Leonard Schibach from Max Planck Psychiatry Munich, we then wanted to dive in and ask again, are there differences between male and female brains? And we wanted to ask this particularly in the context of the social brain, and that is because, according to the social brain hypothesis, complexity in social behavior is, on an anthropological scale or evolutionary scale, a major driving force in why humans may have these super complicated and expensive brains. So within the same species, there may be relationships between complexity in the environment, which is measured here by how many people live in your household, and variability in the brain. So the way we analyzed this is we only looked at volume data from the 36 regions that we already know to be relevant for social interaction in general, as we measured with MRI. And we then applied a probabilistic hierarchical model to get these, what you see here, bar plots, to get like population distributions of how the volume of, for example, the amygdala on the bottom left, how that is different in females in like rows that have less people share their homes with less people, the darker red, more social homes and females living in this environment, and males. So what you see on the bottom left is, it's actually really in females, for example, looking at the social brain overall, is really the more social females that have a larger volume effect in the amygdala, and we don't see that in males. Another example would be socioeconomic status, but that also has a lot of impact on various dimensions of social interaction at like inter-individual and societal scale. And just to give one example, and that is a similar effect in income, how income relates to parts of the social brain in 10,000 people's volume, and healthcare. And in both of these examples, you see that it's actually in males that either have low income or have public health care, two notions of socioeconomic status, they really deviate in their distribution from the rest. So the key point here is we did not want to ask, is there a strict categorical difference between male brain volume or female brain volume? The point of this particular probabilistic hierarchical analysis is to ask, how sure am I that there may be differences in the volume given a social trait, given the population distributions? So, and with this I would like to summarize. We see always more data sets with deep and wide phenotyping at various levels of observation in neuroscience and biomedicine more broadly. And I see this as an opportunity to design and deploy creative modeling strategies that allow us to revisit questions in neuroscience and medicine. And I'm optimistic that statistical pluralism can extend what we can ask about the brain and what we can conclude about the brain. So with this, I thank my collaborators for the inspiration. I thank these institutions for their financial support, and I thank you for the collaboration for coming. Oh.